it. Okay. Okay.
So we've run over a bit in time, so please be succinct with your questions, and we'll proceed with the panel answering those questions. Yes, Skip.
For Dr. Lusis. So it was a beautiful explication of heritable, but right after you described heritable, you jumped to heritable means chromosomal. But we've shown that there are heritable effects, as have many people, of the microbiome. So in your work, I didn't get the basis for believing that the heritable changes were not maternal. And the reason is because the genetics, of course, are biased. I mean, they're inbred strains, so you're breeding them together. So how do you separate, in your analysis, maternal inheritance versus true chromosomal, since it's been demonstrated that there are heritable effects of the microbiome?
Yeah, that's a really good point. I guess I didn't make it
Can
very
you,
clear.
the button?
Mike's not on.
No, make it go.
Here we go. Okay. So, you know, if you look at the, I showed that if you look at the just strains, within a strain, they're very similar. Between strains, they're quite different. That has that problem, right? Because it could be maternal inheritance. When we look at the genetic differences between inbred strains, we're going back 100 years. So if maternal inheritance could persist over 100 years, you're right. I'm assuming that that doesn't occur.
No, I'm just saying that in a facility, when you breed mice together,
Yeah.
it doesn't have to be over 100 years. You're breeding them together. They have the same genes. They have the same microbiome. I'll be happy to talk after.
Well, no, but we don't breed them together. We don't. The inbred strains have been separated for 100 years. They have never been put together.
I think to show that the genes control the microbiome, you have to do the last experiment that you showed, which is to show that when they share a microbiome and then they diverge, then the genes control the microbiome.
Well, you know, I think that that is a good experiment. I totally agree. Look at things all kinds of ways, and I think that that's good science. I agree.
If I can add to that, I think that Jackson Lab mice are re-derived from time to time. There were 2 fires at Jackson Lab when they had to bring the colony back together from users from all over the country and from all over the world. So they were re-derived at that point. So there's not 100 years of difference in microbiota.
No, there is. You know, what happened was that a lot of strains, some strains were lost. But what Jackson Labs did, and this is in the late 40s, there were a couple of fires actually, but they got back strains from the original, you know, they've been distributed to other investors. They didn't reproduce these. There are some strains, like BTBR is more recent. BTBR is 50 years old. But the main strains that we're looking at are genetically. There's a red light. Oh, sorry, genetically diverged 100 years. In my understanding.
All right, let's, with those succinct answers, let's move on to the next question. So yes, who's that?
Hi, I was wondering in the CAFE study if you had any control volunteers who stayed in the hospital for 10 days but were not fed a specific diet, and to what extent you believe that any downregulation of the HPA axis or other factors from not going to work could have influenced those changes in the microbiome.
Yeah, you know, that would have been great. I would have volunteered for that. That's actually a very important point, and that is that just being in the hospital environment could theoretically alter things. So in that particular study, we didn't actually do that. I mean, it's something that we have actually considered, and we're actually digging more deeply now into those types of data sets because we do a number of different types of longitudinal studies. So I really appreciate that comment. It's something we are looking into. My hunch is that that's not the factor because other people have sort of seen something similar to what we've seen. Having said that, I do want to make a pitch. And when doing these types of studies, we very strongly favor longitudinal prospective studies because if we hadn't done this in a longitudinal fashion, we wouldn't have seen any effect at all.
Thank you.
Yes, Karen, please.
I want to return to this question of heritability, and just would like to hear comments from everyone on the panel about what really would be the best trait to measure. So I think there's been a focus on looking at abundance or relative abundance of different taxonomic units, but given some of Gary's data on the striking differences in metabolites, or just the fact that you can have interesting co-dependencies between different members of a community. I think I'd love to hear what are the next directions of what should be the microbiota traits that we try to map.
Oh, okay. So to me, I think there's two kinds of heritability. There's heritability at the sort of species and genus level, which is very high up. And I think this, if we find, there's a decent amount of work showing this, right? But there's sort of these outlier taxes that aren't so frequent. Me personally, I'm most interested in the actual direct transfer. And to do that, we have to look at strains. That we can't, you and I both have an E. coli, we both have a B theta. So, and I think the current methods we're using, unless we switch to deep metagenomics, are not sufficient to do that. But I think knowing what strains can get past one generation, at least if we start to get those probabilities, we can start to extrapolate over future generations. So that's what I would promote. Look in families.
I think Justin Sonnenberg did a really interesting study, I think maybe addresses part of your question as the heritability over time based on diet. And so I think this was published in Nature maybe about a year ago, where you can show that certain bacteria taxa begin to extinguish themselves on if you don't have a lot of fiber fermentable substrate in the diet. And as the whole notion of why our microbiota in industrialized nations is not as rich as the microbiota in a more agrarian culture.
All right.
Oh, just one more second.
Oh,
I'm sorry.
sorry, go ahead, please.
To me, it seems like the major effect, apart from inflammatory bowel disease or something like that, the major effects of microbiota on physiology are through the metabolites they produce. Just based on my limited understanding of, they produce hundreds of different metabolites. We've worked with Stan Hazen on one of these, trimethylamine N oxide, but there are many, many others. And, again, I think that I would look at metabolites. And if I could add a question real quick, or a point in response to Skip's comment, if indeed, what we do is we look at genome versus sharing of microbiota. If maternal inheritance was involved, all it would do is actually decrease our estimate, because it would just blur things. If there's a maternal effect, the relationship between genome and microbiome is going to be, if anything, reduced. Let's have lunch together. Okay. We see something very different. Okay, yeah.
Yes, I think we have some questions from the
Well, actually,
web-based audience.
just one question came in for the web, and I think this is more a philosophical type of thing, so I'm probably ask to limit it to 30 seconds or less. But is to ask the panel to discuss amongst themselves their differences of opinion on diet versus genetics.
Well, I already gave my opinion. As has been pointed out by Gary, that it depends on what kind of diet you're talking about. The diets we showed are very extreme diets. Really high cholesterol, really high fat and stuff, and there's a big effect on microbiota, and that sort of colors my view of things. I really don't know about more modest changes, but I think, to me, if I just had to say, maybe same, 50-50.
I personally think diet in general will have a bigger effect than genetics, but that's just my personal bias. It obviously does depend on diet, but they interact. The influence of diet actually sort of depends a little bit on genetics, and I think we showed that in some of the slides that both of us have shown.
we are very interested in effects of diets that are independent of microbiota. And I think, unfortunately, there's no time to discuss that, but I think it could be a very interesting trend because microbiota is so variable among people. So you have, if you can change biological processes by changing diet that is independent of microbiota, it would be a very interesting achievement.
I do have to add that in our omnivore vegan study, I really think that the majority of things that we actually saw, the vast majority, are independent of the microbiota. These are things that could have been predicted based on just the consumption of nutrients.
Yes, Joe, question?
You know, maybe for Alex, but the whole panel, with the emphasis on accounting for sex in your NIH grants, your applications, your publications going forward, as we look at investigating and interrogating the microbiota, can you expand a little bit about how people should account for sex in their study designs going forward?
I think the simple, for example, doing experiments with SPF NOD mice looking at disease development, there's no point of doing spending money on males because they simply don't develop disease. On the other hand, if we're looking at microbiota influence, then we have to look at males as well. So this is all depends on what exactly you're proposing. So you should say it in the proposal, and the reviewers should be reading what you're saying to get it. And so they wouldn't demand you to repeat experiments if they're not needed.
Thank you.
All right, I want to thank the presenters this morning. Outstanding series of lectures. And now it's time for lunch, and we'll reconvene at 1 o'clock for the remaining session this afternoon. Thanks again.